Okay, show me R, which step you have done. Have you export your results in CSV? Okay, good. Now next step is, okay, please come to ChatGPT and check next step. Give a space or give a line with hashtag. Please press Ctrl Z. Yes, and give a line with hashtag, multiple hashtag for differentiation. Okay, good. Now install this tool. I think you don't have this tool, so first install. Next, you need to wait until installation completing or the red dot disappeared. You need to click here, type here A and press enter button. It will update all dependencies or packages which you need to update. Again, show you a red dot, but you need to again wait for this red dot. When red dot disappear, then you will be able to perform next step. Okay, run next line, library. Yes, it's working. Now come to ChatGPT. Copy second step and run. Paste here and run this line. This ensemble code is for human genes. So our data set is for human genes. So we will proceed for next. If your data set for another genes, you need to mention to ChatGPT, I have this gene sample. You need to give your organism name. So this sample data is for human. So ChatGPT give us a script for human. Okay, now after running, you need to copy next step. Third step, prepare gene IDs, copy, paste here, and run. Next. Okay. Next, retrieve annotation, copy, and run. Okay, next step. Merge annotation with result. Next, next. Okay. Next, copy next step. It's last step. Now come to R and check its result. Yes, you need to, I think you have run this command. Click on file and check gene annotation results. I think not created yet. You need to refresh your screen number 4. Come to screen number 4 and refresh. Yes, no, I think appeared. Click on this file, view. Yes. So here are the results. If you come to your main folder, please open your main folder, minimize R and come to RNA-seq in our main folder, our working directory folder. Yes. No, sorry. Not for GWAS. Yes, RNA-seq. Here are the file, DEG results with annotation. Open this file. It will show you result in Excel sheet. Now each heading and each and everything is clear. Okay, now close and open R. Come to ChatGPT. Okay, now write a message. I have done gene annotation successfully. And for confirmation, take str of your file. No, don't need to write. You can take str if you need, otherwise you can just say I have done. Now your file name is DEG results with annotation or RES annotated. Check in your screen number 3, DEG underscore result. Have you file with the name DEG underscore results? Not. You have file in screen number 4. So first you need to import this file, then you will need to take str. Ignore, come to ChatGPT and send this message. Send this message. No, here is the possible next step. Go and KEGG pathway enrichment analysis. So now we want to perform this step. Okay, copy this step. Step number 1. After creating gene annotation file, you can perform 5 steps. So first we will go for first step. Copy this step. Paste here and write a message. Thanks, no, I want to perform this. Thanks, no, I want to perform this step. So for this, you need some other packages. So first install these packages and then perform all these steps one by one. Please first come to first script, copy and install these packages. Give a line with hashtag for differentiation between above portion and below. Yes. First apply, okay, you can select and run all. Some packages are heavy in size, may take more time for installation. Okay, here you need to update these packages. So come to screen number 2 and type here A and press enter button. It will start updating all required packages and may take more time. Okay, now please all participants perform today till this step, update all packages, and then we will move to in our tomorrow class for Go and enrichment analysis. Okay, now please all participants perform all these steps till to this stage, and please Mozam Ali also share your scripts within Zoom chat as well as in WhatsApp group, and also share your ChatGPT chat. Please come to ChatGPT and convert your all chat into PDF. I think you have free version, maybe not converted. Try to convert. Please convert this. No, not this. Complete chat. Convert all chat in PDF and give me a downloadable file. Okay, download and share this PDF file within group so that participants who are watching recording can also read and understand and can done practice. Okay, thank you. Allah Hafiz.